Welcome everyone. So we are having our weekly seminar at a slightly different time today, and I think it's been worth moving it around because we have a very special visitor, Dr. Amador from Argentina. So Anna's research interests are in biomechanics, neurophysiology, nonlinear dynamics, and more specifically what she's going to talk to us today about, that is birdsong production and perception. So Anna did her undergrad at the University of Buenos Aires, and then she spent, I think, about a year in Brazil, working on the dynamics of epidemics. So she then completed her PhD back in Buenos Aires in physics, where her research focus was on this particular problem, looking at birdsong production. She then won a Human Frontiers Science Program postdoctoral scholarship, which she used to visit the University of Chicago, and her work there resulted in a publication in Nature, which was selected as one of the best papers of 2013. And so that elicited a lot of interest. She then returned to Argentina, where she is now a faculty member at the Department of Physics and a research associate at CONICET, which is the National Scientific and Technical Research Council in Argentina. So off to Anna, and enjoy.
Okay. Well, thank you so much for the invitation and for having me here. It's an honor. So I have plenty of slides, but I don't necessarily have to show them all. So I have a couple stories I want to share with you, especially one beside of the SYN story, the one that I not published. But please stop me, so it will be probably much more interesting if you just stop me for questions and we can maybe discuss and not get even to the 20th slide. It doesn't matter actually. Okay, so my main focus of research is to understand how the brain and the body interacts in order to generate behavior. Behavior in my case is birdsong, and this is particularly interesting because the vocal organ is a nonlinear apparatus. What it means is that with very simple instructions from the brain, you can get very complex output. Okay, so you can be very simple here and yet have a very complex behavior. So this field of research, the interaction between the body and how to get to the behavior, was started by Gabriel Mindlin some time ago. And he studied how the peripheral system produces the song and which are the physical mechanisms involved there. So I had a little part in that story, like when I was doing my PhD. But now I'm more focused in neural coding, and so the idea is to understand the whole path. So now in collaboration with Gabriel Mindlin, that he's mainly more interested in this part, we are generating our own data and trying to understand the whole mechanism. So in our lab, that is the Laboratorio de Sistemas Dinámicos, that is a dynamical systems lab. It's a kind of huge name because you can do anything there, right? So dynamical systems could be anything. So we do some modeling, but we also perform neural recordings, physiological recordings in order to generate behavior and to understand it. So we also record behavior because it's going to be the songbird. Okay, whenever I say behavior, it's going to be that. And the main problem in this is that in biology we don't have theorems. That means that we don't have, we cannot build axioms and from there get to a more complex model. So what I'm trying to say here is that the challenge is how to validate our models. So we generally invent models in order to reproduce the behavior, but the key point is how to validate. Are they wrong? Are they right? Are the simplifications too wide? So that's kind of the questions we have. Okay, so I'm going to tell you a little bit about why we study birdsongs, songbirds, sorry. So why that is an interesting field in research, because I understand maybe it's not so obvious for everyone. And then, as I was telling you before, I will tell a couple of stories of what we've been doing to like the interaction between modeling and the body. Okay, so why we care about birdsong? So songbirds are a great animal model to study complex motor control and the mechanisms of vocal learning. But to stop a little in this, so when the thing is that, or maybe I shall move on. And so there are some shared properties between humans and songbirds. They have similar learning stages and similar sound production mechanisms. So what does this all mean? So when the bird is singing, they are doing something very similar to what we do when we are saying vowel sounds. So if you say A here, you will see the vibration of your vocal folds. And that's the same what the birds are doing when they are singing. So they are not whistling, okay? When we try to imitate the birds, we generally whistle, but that's not what the birds are doing. So that is a very complex mechanism. If you think what you're doing when you are talking, you are controlling your respiratory muscles, the tension in your vocal folds. That's why if you are nervous, maybe your tone just pitch up because you kind of lose control of all the little muscles. And the birds are doing the main thing. So it's a very complex motor control going on just in singing. But on top of that, 40% of the species learn how to sing. So the species that have the more complicated song, they are generally learned. So they go through stages of learning that are very similar to the one of humans. This is they first memorize what they want to copy. They create like an auditory memory of what they want to achieve. That will be a tutor song. And then they start practicing, so it's like a babbling stages when they are always, at the beginning, they always make sounds, like screaming for food, that is kind of innate. But then these screams start getting like a call and they go like getting more and more complex. So that is a sensory motor phase until they get to the final point of having an adult song. And that vocal learning happens really rarely in the animal kingdom. So it happens for whales and dolphins, so the cetaceans, elephants, some bats, and that's it. And 40% of the species of birds. So that's why they are so unique in that way. Okay, so we have a great animal model. I will tell you more about it later. But so birds for us are just like rats in a lab. Okay, they are not, oh, what such cute. No, they are just. Okay, and so one guy is the zebra finch. And let me show you an example. Let me see if I can find my pointer. Sorry. Let's see if this works. Maybe it won't. Oh, that's. Well, sorry about that. We didn't check the speakers. Well, I think it doesn't matter.
I think just the volume was down on the computer.
No, I think it's all the way up. I don't think we have audio.
Looks like the volume.
Yeah.
Yeah, it looks like the volume's just down there on the computer.
That looks like a Mac.
No, because it's not.
It's not going up.
No, it's not going up.
Maybe pull that out.
If you
Play
play just off the Mac. So if you try to get the sound, just
okay.
laptop
Yeah.
rather than-
Oh, there we go. Oh, that's
No, sorry, could I again? No, particular.
No, I think that's the input.
not that one.
But just
Can you try that?
Because maybe it was not.
Very good.
Yeah. Is that what it sounds?
Yeah. Well, I think it doesn't matter much. So.
Is there a sound on the presentation?
I have a speaker I can bring.
Because the thing is that that's okay. It's just a glimpse of that. Yeah, and so what you were hearing, there is a picture here. Okay, it's just like a funny thing, but it's not vital for the presentation. So as it's hard to hear and it's very fast, we use this representation of the song. So these are sonograms. So this is just showing time and frequency, and it's color-coded the energy in the harmonics. So in order to build that, are you familiarized with this kind of representation? Yeah? No? Yeah? Okay. So it's just a Fourier transform doing little small windows and overlapping them across time. So, for example, and so this is a finch song, so it has some particular syllables that are interesting. So, for example, this B syllable or this part of this syllable, if you look just at the window, so this would be the sound wave, it just looks like this, okay? So it looks like a neuron, okay? If I haven't told you this is sound, you would say, well, and I ask you what is this, you would say a neuron spiking all the time. And if you perform a Fourier transform there, you will see the fundamental frequency and all the harmonics, and the higher energy is here, and that is what is represented here or here. Okay, you have higher energy in the higher harmonics. So you have these kind of sounds, and also tonal sounds, that it would be like this sinusoidal shape, where you have one fundamental frequency and almost no energy in the harmonics, okay? So this is the finch song, and I'm going through this in detail because it's just to say that they can be like very complex sounds. Although they are short, this is far less than a second, they could be like very complex. And on top of that, if you record your own voice and see how vowels look like, they look like something like this, okay? So the idea would be if we can reproduce this song, we can reproduce vowels for humans. Okay, so a little bit about how they are producing the song. So the vocal organ is the syrinx, and it's just in there. So in order to sing, the birds just flow air through the bronchi, and they have two pairs of vocal folds. And they put it in a prephonatory position, so the labia are kind of close together, and they set some tension to the labia. And when this pressure is high enough, they start to oscillate. So the way to generate song, and it's the same way we generate the sound of vowels, and this is the last time I'm saying that, is just with the perturbation of the air. Okay, so if you have this movement 400 times per second, you have a sound of 400 hertz. Okay, like a la, like a, how do you say la? In what? In music?
A note line.
A.
No, just
A, that's it. 440, so that would be A. So the way to generate an A, it would be like to put in oscillation. So that's why they can generate, for example, these high notes that are almost 5 kilohertz. And it's not because they are having a muscle moving at 5 kilohertz. It's just that putting a lot of tension in the labia, and then the air is flowing, and the more tense it is, the more higher pitch you get, like in a guitar. And so in that way, these auto-sustained oscillations are the ones that are generating the sound. Okay, and then they can control one side or the other one, and in that way they can make different sounds, but we will concentrate just in one part. Okay, so you need syringeal muscles that are going to be the muscles that control this labia, and respiratory muscles, you need to coordinate them in order to generate the sound. So, what does a physicist do with all this information? It just gets things simpler. So you have a very complex biology, a lot of muscles, but at the end, you have some labia oscillating. So for a physicist, if you have some labia oscillating, you will just have a spring, and that's it, okay? So this has been work from Gabriel Mindlin. So he started with a much more complex model, like he started from the physicist's point of view, trying to describe all the details a physicist can, that it's much less detailed than in a biology would like, okay? So from biology you have thousands of details, and a physicist what will do is will just try to pick the more important aspects. And so after several years of research, we end up with this model that is just so this part here is just a spring. So if I wouldn't have all these nonlinear terms, this part would be just a spring with an applied force. And so the interesting thing about this is that all these nonlinear terms were a deduction from the physical model. If you're interested about it, I can tell you more how we did that deduction after the talk. But the interesting thing about this model is that, well, X is the departure from the rest position of the labia. So whenever X is oscillating, you will have sound, okay? And if X is fixed, if you have a fixed value of X, you won't be producing any sound. And the interesting thing of this model is that you have just two parameters that are related to things that we can measure. So these are pressure of the air sac system. So the birds have a fixed lung and balloons connected to the lungs. And with this, so the lungs are kind of fixed. They are not expanding like ours. And these balloons pump air. So this air sac system is the responsible of getting this jet. And the tension of the syringeal labia is the other parameter that is important, and then gamma is just a scale factor. Okay, so we have this model, and with the two parameters that we know what they...
What are the rationale to have only gamma in different powers at different... So you have one parameter with scaling factor.
Yes.
Are there any theoretical considerations why it's only one parameter in all these terms?
Why just one scaling factor and not more?
Yeah.
Why not?
Yeah, you have one, it's gamma squared, then you have gamma without square, gamma without square, gamma squared, because it's probably the...
Yeah,
yes,
so this gamma or gamma
so
squared...
this part of this just dimensions, but why it is only gamma?
I'm not sure I understand.
Yeah, if you write the general equation of all these terms,
Yes.
you would put
So
that for each you would have each particular scaling factor.
Yeah. So the thing is like this. We started with a physical system, and then this is the normal form reduction of that particular system. Okay. And so that is why, and that is also why not also the gamma. The other question would be why keeping this nonlinear term and why not y to the third, for example.
Yeah.
Well, that answer is just in the dynamical analysis, and I can tell you more about it after the talk. So,
Okay,
what
some
we
rules
did
from some physics, right?
So so it's more or less a dynamical analysis. So we had some dynamics that we want to reproduce with less parameters. So we started with less weird terms here and just deducing the terms from the physics. But the problem with that other model is that we have so many parameters. Okay, so in order to reduce the parameters, we did this normal form reduction that is just a technique from nonlinear dynamics, and then that's why these kind of weird terms appear. But we can maybe discuss it later. Okay. Okay, so with this, so you have just to trust me that this is a good model and that you have like these two meaningful parameters there. And so this is also part of the dynamical analysis of the model. So you have this alpha parameter and the beta parameter that again relates to things that we can measure. And so here is what you have. So in this region of parameters, so if alpha is 0.1 and beta is 0.2, you will be like here. So in this region you will be oscillating. So this is a limit cycle. So this means that x is oscillating. So in this regime you have sound. In this regime, for example, you have a fixed point, and so that means x is going to have a fixed value, and so it's going to be silence. And this is more like an excitable regime. So you have a fixed point, a saddle. So this is more like a nonlinear dynamics analysis, but it's just telling you what the variables are going to do. So in this place, you will get also to a fixed point, but with an excitable pattern. So that would be that if you are close to here, you will just end up to a fixed point, or you can make a long excursion. So here you will have a region that can represent neurons. Okay. So these lines are what is called bifurcation lines. That what it's doing is separating qualitative behavior. So here you have a fixed point, so silence, and here you will have sound. And again, this is another bifurcation, and you will have silence here and sound here. So the interesting thing there that it depends how you cross from silence to sound, you can generate different sounds. So if you cross it through here, so this is the value of alpha, so you increase and decrease, so you will be doing increasing and decreasing. If you do it through here, you will have, as you are going through this excitable regime, here you will have oscillations, but you have still the presence of this neuron-like excitable regime. So you will have a lot of energy in the harmonics. If you go from silence to here, from here to here, you will have then more like a tonal sound, okay? So this is just to tell you that changing, so if you just make path in the changing in alpha and beta, you can generate sound, okay? So that is what we did. So actually this is the actual integration of the model. So this is alpha and beta, and these are the paths that we took. And here you can see this is a recorded song, and this is a synthetic song. Let me see if you can hear about it. This is not so easy. Okay, that's the zebra finch. Yeah, I'm sorry, that's all we have. So as you hardly hear it, you will feel that this is great. Okay, this is the recorded song. So here you can see the sonogram, so you have the recorded and the copy. Okay, so that's the idea. Changing alpha and beta in a given way, you can generate a synthetic song. Okay?
Yeah, alpha and beta are slowly changing. Right?
Slowly changing, right?
Yeah.
That's important because they are much more slower than X. Because X is oscillating and it's like 400 hertz.
Yeah, so all analysis is basically alpha and beta constant.
Yes, yes. So all the analysis that you have like a steady value there, and you have a very, so it's X is much faster than alpha and beta. Yeah.
So that second sound rate, that's a synthetically generated.
Yeah. Yeah.
It sounded exactly the same as the zebra.
Yeah. It's kind of a trick because you are hardly hearing it. So it was all planned, this thing of not having the speakers. No, it's really similar, and especially like the picture also are really similar. Okay. So yeah, so we have this model that can sing. And so the question here was, what can we learn from this? So here it comes two stories.
Sorry, if I could go back to that. So was that a matter of using alpha and beta as fitting parameters to
Yes.
to match the...
Yeah, so what we did was matching the fundamental frequency. We tried to match two values that we extracted from the sound. The fundamental frequency, and then we developed what we call a spectral index. It measures how complex the signal, spectrally complex the signal is. Yeah, so we matched like these two features that we extract from the sound, and then we feed these parameters. We are not claiming these paths are unique. They are just in a region that we kind of like. And for all the birds, we can have a kind of common place where to move those parameters, okay? Because each bird has this unique song, okay? So each bird is singing its own syllables.
And what role does gamma play in all this?
So you can make it a higher pitch or a lower pitch, if you will. Yeah. So for the same alpha and beta, yeah, you can change the pitch. So we just fit the gamma and then just leave it there for all the species. But also even changing species from zebra finches to canaries, we didn't move that gamma factor. It's just there. Yes.
It just scales.
Yes.
It's uniformly. It can divide by gamma squared.
Yeah. So it's just scale. Yeah. It's just there. Yeah.
You were able to measure the tension and the pressure in the bird.
Yeah. So pressure, yes. I mean, well, it's also. So we can measure pressure more directly. We can measure the pressure in the air sacs, although this pressure would be just the pressure in the vocal folds, and that could be a scaling factor there. But that is the tricky one is the tension.
Sure.
Because for set the tension, the bird has several muscles. So we can measure activity in some muscles. So I didn't do that, but they were measuring with some collaborator. They were measuring activity in the muscles, and then feed that as a parameter, and they could reproduce a cardinal song, for example. So for simple songs, that works. So they feed pressure and this activity of the muscles, and they can have this reproduction. Yes.
By tension, what you model, not what you measure, I imagine what you're modeling the tension along the edge of the leaflet of the cord. Is that correct, what you mean by tension?
Yes.
Thank you.
Yes. And that's why that is a very indirect measurement, what we measure in the muscles. So we know there are some muscles that are more related to fundamental frequency in the sense that if they change, they were measuring the tension and recording the song, and they could relate that if there were more activity in those particular muscles, the pitcher would go high. So they correlate the tension and the fundamental frequency. Well, okay, so in between should be tension, but that is for particular birds, for particular syllables. So for simple cases that work. For more complex, it's more tricky, but still we can make a simplification. Say, well, we cannot measure the tension, but if this is the tension that is achieved, you will have like this sound. But maybe this tension could be hard to get, or the activity of several muscles. Okay, but we still need to work on that. We would need to measure several muscles at the same time, and make an integration, and to see if we need to improve the model. But what we did, say, well, okay, this model have some prediction. So this is the same picture that I was showing you before. So this is silence, and this region is sound. So here what I'm plotting, so here I calculated with the model what value of the fundamental frequency you will have if you are in this point of the parameter. So with this value of tension and this value of pressure, what would be the oscillation like? Okay, so one prediction is that if you increase the tension, you will increase the fundamental frequency of the sound. That is what I was telling you about. This we needed to achieve because this has been measured before us. So the fundamental frequency is controlled with the membrane tension. That is something our model can reproduce. But the other new thing about it is that if we go the other way, so we fix the value of tension here and we make this cut, so we increase the value of pressure here, we can also increase the fundamental frequency with pressure, okay? So that is one prediction of the model that was kind of weird, okay? So as thinking in this way, if you speak louder, you will increase with a higher, you will talk with a higher pitch. That's our prediction. And that's what generally happens. The only thing is that when you are screaming like me, what you are doing is controlling your muscles of the vocal cords so that the pitch remains at a constant value. Okay, but so this was a new prediction from the model. So we wanted to, and this was also a novel mechanism for vocal control, that you can control fundamental frequency with your tension or with your pressure, okay? And we wanted to see if we can find this in the bird. Okay, so this is a zebra finch singing again. And we can measure pressure while the bird is singing. So we put a cannula in the air sacs and a pressure sensor in the backpack, and they are there, so happy singing. And so these are the syllables are singing an expiratory pulse, and you can see you have like different shapes of the pulses. So what we wanted to do was to kind of manipulate the pressure here, okay? So our goal was, well, look here, you have steady pressure and steady fundamental frequency. So our idea was, if we can manipulate this pressure while the bird is singing, if it is true what our model is saying, if the pressure goes down, the fundamental frequency should go down, okay? If the bird is not compensating. If we depressurize and this keeps steady, we cannot say anything, okay? Because it can be that the bird is compensating, we don't know. So it was kind of a crazy experiment, those that you do in your postdoc. I don't know why, I mean, because it's not a way. Okay. So we built a valve. Here you can see the bird and with a valve in the backpack. So this valve, we just put a cannula in the air sac, and here we have a rare earth magnet, a solenoid, and a metallic, just like a metallic plate. So if you are not passing currents, the magnet will just stay in this metallic plate, so it will be closed. Okay, so imagine that this goes to the air sac system, and this is open to the atmosphere. Okay, so if the magnet is here, it's closed. But if you put some current through the solenoid, it will move the magnet in this direction, and so the air could pass through the valve. Okay, so you will have no current. And then you will have, when you give a pulse, it will be open, and then you give a little pulse to close it. Okay, so with short pulses in one direction or the other, you move the magnet. Okay, so we have to build, so I have to build it because we couldn't get something commercial that is small enough. So these birds are like 14 grams, so they are really small. And everything is humid, okay? The air is really, it's breathing. So it's really bad. Okay, so we have this valve and some wires going up, so I can open and close the valve when the bird is singing. So that's what we did. So, well, here I target this one that has the same thing, so steady pressure and steady fundamental frequency. And when I open the valve, the fundamental frequency just drops, okay? So that was what we were expecting, especially you can see it here in the higher harmonics. So this is what is showing is that exactly what the model predicted, okay? That we can control the fundamental frequency with the pressure, okay? And the other thing is that we know that the birds are not compensating. So it has to be subtle, okay? So if we just gave like a beep, so if they depressurize very fast, they kind of run out of air and they would stop singing, okay? But if you are gentle, they will just continue trying to sing, but they wouldn't compensate.
There looks to be an abnormality, and if you look at C prime there, you can see the higher harmonics fall off more than the lower harmonics, which seems anomalous.
But that is just because it's harder to see. So so you have like a gentle drop, okay? It's because you have a gentle drop in the pressure, and so the thing is that if you look at these harmonics, you will see just a gentle drop, but you will have to like zoom in, okay? In the harmonics, this is just amplified. So it's just because you're just multiplying, okay? So if you are...
You probably want to write on the other side.
I'm so sorry!
That's okay.
What happened? So the idea would be, for example, this is the fundamental frequency, okay? And if you are looking at the 10 harmonic, you will have like this number, okay? So if you just have a variation that it will drop to 3, so it will be just drop a little bit, just 20 hertz difference, but here you will see like 200 hertz differences. And so it's just, so we are just looking here because it's easier to see, but it's not that it's not there in the fundamental frequency, okay? It's just, yeah, it's a trick. Okay, I'm so sorry, it's so late already. Okay, so this is one result. So we had, so this is kind of the work we do. We have a model, but we make a prediction, and then we try to put our model on text, okay, and see if we can learn something about it. Okay, with this model, we learn that it is true that the birds can use the pressure in order to control the fundamental frequency. Okay, so this is just a sum of that. So what we measure and what we model, and it's the same thing, but I just wanted to tell you another story. That is, okay, so we have a better understanding of the mechanisms of song production, but the question at the end is, how good is the model? So we know it's good that we can make some predictions, but the question is, how good is this copy? So for that, we are going to use some neurons that are particular. So this is a picture of a bird brain. And the interesting thing about this is that they, in some, so these are neural nuclei, so a bunch of neurons together, and they are completely dedicated for song production and learning. And there are some neurons here that will just fire to the song presentation. So the bird's own song, okay? So each bird has its own song, and when they are singing, they have a particular pattern of firing, but if you play back the birdsong song, these neurons will fire in a particular way. So this I won't show you. If you want to see some movies about the birds, because this won't play nicely because you need to hear. Okay, so that is the bird. Each click is a neuron, and when the bird is singing, you can hear what the neurons are doing. Okay, and it changes its pattern of response when the bird is singing. So we have these neurons, and that would happen, and if they are just hearing, that happens in the same way. So for example, well, let's see. Okay, well, it doesn't matter. Here the bird is sleeping. Okay, I don't know if you can hear that, but the pattern is changing when you're presenting the birdsong song. And the interesting thing is that only happens, so it's not sound. If you play back another sound, the neurons won't fire at all. So you can see that. So this is how you represent that. So this is a raster plot. So each point is a spike, and this is time, and this is number of presentations. So what we are doing here is that we are just presenting the birdsong song here, and what we see is that during this presentation the neurons fire a lot, and it's completely different what happens here than what happens here when they are not hearing anything. Okay, so this increase in the firing rate just happens when we are presenting the birdsong song, but if you present other songs like the reverse song or a conspecific, what happened was that they won't fire at all. Okay, so this is what we call selective neurons, in a way that it's very hard to get them to fire. They will just fire to the bird song. Okay, so in this way, so again we had the recorded song and the model, and what we wanted to do was using these neurons. Like, is this copy really good? If it's good, the neurons would fire in the same way as when we are presenting the BOS or we are presenting the synthetic. And if it's not good, it won't fire at all, because it's really hard to get these neurons fire. Okay? Okay, so we did the experiment, and so get what happened the first time we did it. So the first time we did it, we didn't see anything, okay? This was a failure, like a total failure. So if the model would have been right, I mean, so this copy that we liked so much before, when we did the experiment, the neurons told us, like, your model is not good enough, okay? So to you it can be a kind of a nice copy, but the bird, it was the same. The synthetic was the same as a conspecific, like whatever bird singing around. So the neurons were telling us the model was not good enough, okay? And so that is unpublished results, obviously. So the problem there was how to continue. Again, the problem was when you have these problems in biology, it's hard to know where to go, because if this would be mathematics, you say, well, I will just increase the order of the approximation. Maybe the second order was not good enough, I will go to third order and all that. But in biology, you don't, I mean, you are always not considering a lot of details, okay? So the question was, well, how to not increase so much in complexity, but still get a good copy. So what we did was we look and hear a lot of songs and synthetic songs, and what we notice is that the real song is much more noisier than the one we had. And also you can see a band, so these harmonics are, remember that this color is higher energy in the harmonics, and this is more like flat, okay? So we increase the complexity, adding noise, and adding this band. So we keep this model for the sound source, but we added more complexity to the filters, okay? So I will just speed up here, and I can tell you more about it later. But at the end, what we got was a more similar song. So now it's more noisy and with a band. But now we wouldn't believe to our criteria, okay? So to us, this was great, but before doing the experiment, this was great too. Okay, so we did the experiment. Now again, we have another song, another neuron, because each neuron would fire in a different way, but the same features. So if you play a reverse, you wouldn't see a response. If you play conspecific, you wouldn't see this excitatory response. And we finally got our synthetic song that would fire in the same way as the Birdsong song, okay? So this was the great success. So it's the same pattern, maybe with lower amplitude, okay? The real one has like more probability of firing, but the key thing is that when this neuron fires, it will fire in the exact same place, okay? So this was a great result, so we did it more clean. So we now play back 3 times the Birdsong song, that is the way they generally sing. And each time the neuron will fire in a particular place, and then the synthetic song is doing the same thing, okay? So this was a great thing for us, for physicists, and we were so ready, so I was so ready to go back home from my postdoc and say, well, as a physicist, you have a low-dimensional system that is very simple, and yet you get to reproduce the complexity of what a neuron is doing. But my boss, at that time, Dan Margolias, said, I don't care about your model, because you have a nice model, but I have another friend physicist that have another model with 100 variables, and maybe it's better that model than your model. What we have to do here is that your model tells us something about what is happening. So we have to use your model as a tool. It's not that the objective a model. It has to be a tool. And that is where interdisciplinary works really gets interesting, okay? Because you have like different views of what a problem is or where the success is. So this is just a detail. So I will just stop here, kind of just leaving you with the idea that now, instead of looking at this very complex signal, we are going to be looking at these very much simpler signals. And this is what we call the motor gestures. And what we unveil looking at these gestures is that the neurons were not firing in every place. They were firing at particular instances that maybe in the sound were difficult to look, but when you look at the gestures, these gestures, for example here, you can see that it's a transition between gesture and the neuron were just firing here. So these are just so this is time, and this is like the neurons firing. So with this model as a tool, we propose a new way of coding for neurons in this neural nucleus, that is that they are firing at particular motor instances. And this is kind of new, because until we arrived, the model was that the neurons were firing all over the place, and we are proposing that they are firing just at particular motor instances. So this has been a big fight in the area. People are just struggling. Is this model? We're just making now experiments to see who is right, but it has been an interesting trip. So we are doing experiments right now. Yeah, but I just would like to finish with the conclusion. So we use a low-dimensional model that can elicit complex neural responses, and the importance of dimensionality reductions to study complex behaviors. And especially, it's important that you can relate the parameters of the model with things that you can measure so that you can validate your model. And once you have that, you can study motor control, and in this way propose novel neural code for brain areas. So I think that would be all. Thank you so much for your attention.
Thank you, Anna.
Okay.
We have time for a few questions. Any questions?
Yes, presumably when the bird is singing, those neurons that fire when it's playing, when you're playing the birdsong song, they also fire during the production of the song. Do they fire at exactly the same phase in relation to the song as they do when they're receiving auditory input?
Yeah, so that is a tricky question. It depends on the species. So there are some experiments, very beautiful experiments done in sparrows and Bengalese finches, that are some birds that are very territorial. So they will sing if another bird is singing in the same territory. And so they were recording the song while they were recording at the neurons. And then they play back the Birdsong song, but then the bird will think there is another bird and they will start singing. So in those experiments, they can hold a cell, a neuron, and they will record while hearing and while singing. That's your precise answer. So the neurons fire at the exact same place. The difference is that when they are singing, it's more like a burst. And when they are hearing, it's just one spike or one or two spikes. But it is really interesting that it's in the same phase. We are doing those experiments in Canaries right now, and it's not so clear to us. So probably I will get back to you in a couple of months to tell you about it. If they are anesthetized instead of just hearing, it looks like there is, so in anesthetized they also fire. In anesthetized it looks like there is a phase difference, but still we need to look at more data. But this thing of having the same phase is very interesting. So they call those like mirroring neurons, because they were doing the same firing when hearing than when doing the action, okay? So they are premotor, but not in the same sense that premotor, that it's like a domino change until the motor action is. So these neurons are premotor in the sense that if you lesion this neural nuclei, the bird won't sing. But this synchronization, having the same phase when hearing and when producing the sound, that is telling you that something else is going on there. So our idea is that you have what we design like what we call it, like a circular model. So everyone is looking this like a top-down, so from HVC to the respiratory nuclei. But we know through actually a lot of work done by Martin Wild that there are some other connections going up to the system. So now our claim is that there is some synchronization going in this circular architecture that allows you to fire in the same phase. But still we are working on it. But yeah.
Any other questions?
Why is the bird's own song so important to it? One could imagine in survival terms, the songs of conspecifics or the songs of predators or something, other things would be much more important.
Yeah, so these neurons in particular, it's kind of, this nuclei here receives auditory inputs, and it's like the first well, there is, let me put it in this way. This neuron receives auditory inputs and has motor outputs, okay? So, so this selectivity firing and at its own song, this property emerges when they are learning. So it's a combination between auditory and motor. So this part, so this new, and this happening in this, and once these properties develop here, then it's translated to the whole network, okay? But I think it's more like a thing that is in this neuron, it's receiving auditory and it's getting the outputs of motor. So I think it's kind of a, we think it, but still we are thinking on it. So it's like like you have to match different, so you have to match the auditory and the motor in order to learn. I mean, in sensory motor learning, you have to match what you are producing, what would you expect. And these neurons in this nuclei would fire when hearing and when singing. So I think it's a particular code.
It's a curious thing. Your example of the bird's neuron firing while it was asleep was remarkable. To recognize your own song while you were asleep implies that it has some spatial value, almost beyond a learning procedure.
Well, I mean, it has to learn. So the thing is that this property emerges while learning. The more ethological, like behavioral side could be that these birds lives in colonies, so it can be like 100 birds colonies and all that. And so, and it's kind of, everyone is singing its different song. So its own song can be probably to like differentiate between others, because like
But it would never hear its own song sung while it's in a colony.
Well, when he sings.
When it was asleep, yes, I see.
Yeah, so
Yes.
when he's singing, he will hear his own song, and that will kind of separate between all the other ones.
Yeah.
But I don't know why it's so important in biology. It's kind of a weird question. So yeah. So I don't know.
It's important for song production because you need to have neuron which is tuned to your own song to be able to correctly produce it.
Yes. So what happened with these birds is that if you deafen them, so if they cannot hear them singing, they kind of degrade the song. That is the same thing that happens with humans. If you have an accident and you cannot hear it anymore, then you start to degrade how you talk. And with birds happen the same thing. So they will stay stable for some time, but if they don't have the auditory feedback, like just the motor output from here is not enough. You need to have some feedback, and probably that's where it's happening. But why is it important during sleep? That is also speculative, but the idea is that probably they are learning or maintaining its own song during sleep. This kind of replay during singing, during sleeping, we've seen that they do that during sleep. So during sleep, if you're recording neurons here, you can see the same pattern as that you saw when they were singing. So they are kind of having a replay of the song during sleep. Why?
Isn't it weaker then? Is it exactly the same as an awake animal? Or is it still weakly?
Well, that's a great question. It is not exactly the same. You can still see the pattern, but sometimes it shrink a lot, or sometimes it gets a little bit longer. So the timing is not so precise. It can change a little bit the timing, but the pattern is kind of complex, and so you can see still there.
Fluctuations in temperature.
Yeah, that too.
It would believe the cooling experiment.
Yeah. So what Martin is talking about is that if you cool the brain, so if you put a Peltier and pass current, so everything is cooled down, the birds start to sing more slowly. and presumably the neurons will start like firing with a different timing. So yeah, that's fine.
Right. Well, the last
Sorry, did you analyze the dimensionality of just the raw sounds that were produced by an individual bird?
Actually, no, because we think like, we thought it would be like too complex, so we didn't do that. But
While the sound wave is very complex, if the bird, an individual bird, produces exactly the same sound wave every time, then that's a zero-dimensional system. But if there is some variation, it may be in pitch, it may be in the duration, or it may be a combination of, any of the very high dimensionality signals or parts of the signals that you acquire. That will give you, if you do, say, for instance, principal component analysis of that will give you information about the dimensionality of the system that is producing that complicated sound. And it may be very low dimensional, in which case, that might give you some assurance that the low dimensionality model that you have got is valid. Or it may be relatively high dimensional, enabling the bird, for instance, to have be able to communicate more richly. And if the dimensionality is higher than the parameters of your model, then you've got problems.
okay, so yeah, we know there are some subtle variations in fundamental frequency and in duration. For example, you have more variations if the bird is singing to his own, but if he's directing the song to a female, it's much less variable, okay? But we know there are some variations. They are both in fundamental frequency and in duration. Spectral analysis have never been done to there, so it can be interesting to look. I mean, we have never analyzed the variations from a recording of the whole song of the bird. So also what is important is that you have the song, but then you have calls. So song is mainly for mating and for marking the territory, but you have then the calls that are like short syllables, but they mostly communicate with those. So you have like 10 or 15 different type of calls with different meanings. So they perform this call to like an alarm call, or to say, well, there is this man coming through in the room, they would just do experiment to us, or something like that. So they mostly transmit information using calls and not necessarily song. But what you're saying about analyzing and try to kind of estimate the dimensionality with the sound, we never try it. We can try something along that direction. The problem is that what we look is that it's a very complex signal. So I don't know. And for some syllables, it can be like a very complex spectral thing. But I don't know.
But it's not too complex,
It's not too complex. The thing is, even if you with PCA, you find that it's a high dimensional system. It doesn't exclude that in reality it is a low dimensional system, because it can be non-linear. It can be some kind of non-linear surface in this multidimensional space. And the other thing, that the two parameters, they are functions of time. So strictly speaking, both are infinitely dimensional parameters.
Yeah, that's right.
But for short periods, as far as I understand, you have constant, and then you have two-dimensional, and this gives you for a short time, you have a particular pattern.
If you have a system that has, a parameter that can vary with time, that is a two-dimensional system. It's not an infinite dimensional system.
Yeah, right. Assuming, of course, if these parameters, if the variation in time of these parameters, if these parameters can be assumed to be constant at this.
No. They a particular I suppose, output of that system may be a curve that goes with time and the variation of that single parameter with time.
Yeah.
So that is a two-dimensional response. There's only two things that are varying there.
Yeah, but time is also a parameter here.
Yeah, that's what I'm
So
saying.
we have a
We've got
time of-
one parameter that is varying
if
with
all
time.
the variations are very fast compared to the variation of A and T and A alpha and beta, then yes. But as soon as alpha and beta start to change fast, then it's both become that both alpha and beta become a infinitely dimensional parameter because it's a function.
Okay.
I think that
Maybe we can talk it over.
I think it is. I think it is two-dimensional.
Yes.
It's right.
all right. So let's thank Anna and
Thank
move
you.
over to the
Okay.
refreshments.